For today's lab, we're going to be talking about nitrate and ammonium extracts. So nitrogen is a super important nutrient, super important part of the environment, but it's also tricky because it's everywhere, right? In the air as a gas, it can be a solid or it can be dissolved in liquids, and it has a bunch of different forms or redox states. So you've got nitrogen gas, which is N2, that has a redox state of 0. It can go from a -3 or up to a +5. So one of the most important forms of nitrogen is inorganic nitrogen, and that's usually nitrate and ammonium. In order to measure nitrate and ammonium in plants and soils, we're going to use an extraction. So I've set up a table here to kind of just help you think through this. So we've got, you can directly run, you can extract, or you can digest, and the different materials or matrix, the soil, plant, and water. Now, if you're measuring the total nitrogen, you're going to run that directly on the sample. If you're going to run nitrate and ammonium, you're going to do an extraction for soil and plant. For water, you're going to filter it because you can't extract water, it's already a liquid. But this extraction process is going to take the soil and the plant, and it's going to turn it into a liquid that we can run on the instrument. Now, it's not going to completely dissolve all of the tissue. That would be a digestion. That's what we do in the microwave. And that's what we would do for measuring the ICP elements on each of the different sample types. There's also a difference. Some of these extractions will run a total measurement, and some of them just do what's plant available. So in green here for soil, you get the plant minerals when you do an extraction on a soil. But when you do nitrate and ammonium on soil, this extraction actually gets a total nitrate measurement. So we're going to do an extraction, it's going to remove all of those inorganic nitrogen so that we can measure it on the instrument. So here's an overview of the procedure that we're going to be doing today. So previously to this point, we have collected the sample, we've shipped the sample to the lab, it's been dried and ground, so it's now ready for analysis. So we're going to do the extraction and then set up the instrument, run the samples, and then review the data, do any QA/QC at that point, and that's what we'll do for today's lab. We're going to briefly go over how the extraction works for the plant and for the soil. The water samples would just be filtered and run directly on the instrument. But for the plants, what you're going to do is you're going to weigh out some of the sample, so make sure it's mixed up. We're going to weigh out 0.25 grams into a tube. A little tear here. Too much. There we go. So we've got 0.25 grams, and we're going to put that into our tube. Get as much of that in as possible. And it doesn't have to be a quantitative transfer. Just dust that off, and it's ready to get the next sample. And then what we're going to do is we're going to add 25 milliliters of the extraction solution, which is 2% acetic acid, into the vial. And then once that's in there, we'll cap the sample, and then that's going to shake on the shaker for 20 minutes. Now for the soils, we're going to weigh out 2.5 grams. So we'll clean off spatula between samples here. Clean out our weigh boat. We're going to tare it, homogenize this, and grab 2.5 grams. A little more accurate. There we go. Oh, it's fluctuating a little bit. It's okay, we just want it to be plus or minus 0.01. So that's close enough. So we're going to put that into a tube. Now for soil, instead of the acetic acid, we use a KCl extraction. So this is the salt is going to break any bonds between the nitrate and the ammonium in the soil particles. And it's going to rupture any cells that are in here as well. So that's going to put all of the nitrate and ammonium into solution. So we'll just use this auto pipetter. So we just need to pull all the way to the top, check that it's set to 25 milliliters, and then we can just do that into here. And then we'll cap it. So the plants shake for 20 minutes, and then the soils will shake for 1 hour. Because this is a total nitrate and ammonium procedure, shaking for a little bit longer won't affect how much of the nutrient is released into the solution. It should release all of it, so it should be done pretty quick. There are some extractions that are for soils that are the plant available nutrients, so those are a little bit more time sensitive. But we'll follow the recommended procedure here. So once those are all weighed out, make sure you do a blank and a duplicate in your KC. We're ready to go put it on the shaker. Once those are all in your tubes, we can put them on the shaker. So we wouldn't want to mix the plant and the soil samples since they need to extract for different amounts of times. But we're just going to show you an example of what that will look like. So you'll put this into the shaker, and then you're going to adjust this bar so that it secures those samples in so that they're not moving around too much. Once that's tight, you go ahead and put the shaker on low. And make sure that the lids are screwed on tight so that they're not leaking. And we'll let that shake until it's done. And then we'll pretend like it's been the amount of time. All right, once the samples are done shaking, we'll come back to the shaker. We're going to turn it off. And we'll reverse the process, loosen the bar, pull the samples out. So these samples now need to be filtered or centrifuged and then filtered. Centrifuge is over here. The instructions for using the centrifuge are on the wall. But one of the most important things is to make sure that you balance the centrifuge. So if you have an even number of tubes, that's great. You can just put them in. If there's an odd number of tubes, you don't need to centrifuge the blank. Or you can add a water sample so that you have an even number. The slots are numbered, so we'll just start in 1 and 2. Close the lid, rotate the knob to lock it. We're going to set the timer for 5 minutes, and then we're going to turn on the speed. We're not going to go over this duct tape line. Okay, when the centrifuge finishes, the timer will be at 0. We're going to come over here. You can use the brake to slow down the spinning. Just pulse it so it doesn't wear out the brake. You can hear it slowing down. Once it's finished spinning, we'll just turn the speed to 0, and then this will be able to be unlocked. So we can push this button. Kind of hear it. All right, once we get that click, that means that it's unlocked. We can open this knob and pull out the samples. This one has a little bit of color in it. So we didn't add any activated carbon. So what we would need to do is re-extract this, add a little bit of activated carbon to make sure that this solution is clear. We don't want it to be gray from the activated carbon. So sometimes you kind of have to redo it a couple times until you get it right. And we usually don't do any to start with, and then if it does have color, we'll re-extract it. But that's the plant samples. The soil, you won't need to add anything. They'll be clear and they're good to go. So we'll come over here to set up our Nalgenes. The Nalgenes we keep in this cupboard here. And we'll use these smaller sizes. And we're going to filter them with filters. We're going to use the 413 12.5 centimeter filter. And we're going to fold it in quarter and then open it, back fold it, and notch it. This way the filter is going to sit in the bottle really nicely. So we'll take the sample. These filters are big enough that you can just pour the whole sample in. And it will have space and it won't overtop the filter. We want to check and make sure that there's gaps between the filter and the edge of the bottle. If there's not a gap there, positive pressure will build up inside the bottle, and instead of water filtering through the filter into the bottle, it will start coming through the pores on the side and overflowing onto the counter. That'll make a big mess, and we don't want that. We'll do the same thing for the plant extract with the acetic acid. Fold the filter in quarter and then notch it so that it doesn't seal against the side of the bottle mouth, and then pour the sample in and let it filter. When it's finished filtering, you can take the filter and it can just be thrown away. And then the caps are up on this shelf above. So we'll get a cap. You can cap it. This can be stored in the fridge for about a week while you're waiting to run it on the Feolizer for the nitrate. If you're running the soil for ammonium, then it needs to be run within 24 hours. Once it's done filtering, you can put it on an ICP rack. Make sure you've labeled out all the spots so you know which samples went into which test tube, and then it will be ready to run on the FIA. These need to be washed, and we don't want the soil pellet to dry in here because it'll make it harder to get out. So once you're done, take these over to the sink, scrape out the pellet into either the paste bucket or into the garbage, and then this will be washed with hydrochloric acid and dried on the drying rack. Now that we've got the extract in these containers, we can put it onto an ICP rack. Make sure that you fill out your ICP rack map to record where all of the different samples are. And then we're going to run it on the FIA. This is the FIA. That stands for Flow Injection Analysis. This instrument can run nitrate, nitrite, ammonium, and SRP. We're not going to talk about SRP today, but we'll talk about those nitrogen. So the way that it measures nitrate and nitrite is using this pump. The method is called the cadmium reduction method. The way it works is that the autosampler takes up the sample and then it mixes with sample that comes in through these other with reagents that come in through these other tubes, mixes them together, puts them through this reduction column, and turns all of the nitrate in the sample to nitrite. Then color develops as it goes through this mixing coil, and the color is measured as it goes through this flow cell by a spectrophotometer, and then the rest goes into the waste line. And the waste goes into a waste container down here. The way that ammonium is measured is using an IPB, that's indophenyl blue, salicylate method. Similarly, it takes the sample in, mixes the sample with reagents coming from the sample tubes, and puts it through a flow cell that measures the color blue development and records it on the spectrophotometer. This one doesn't have a reduction column. It does have a heating cell that changes the temperature in the coil here so that the color development is really consistent, because the color development is temperature dependent. So flow injection analysis, basically it's an automated spectrophotometer. It pulls in the reagents. The injection system here in the middle adds the sample to the reagents, and then it measures on the spectrophotometer. The system requires regular maintenance, so we have to replace the tubing, the different parts in here, the flow cell, the cadmium column, and something else that's really important about this instrument is making sure that the samples that you have, the calibration standards you run with each run match that matrix. So for example, for the plant nitrate, we've got nitrate in acetic acid. Depending on the ammonium that you're running, you can do nitrate and ammonium in water, or you can do them in KCl for the soil. And then if you're running nitrite, you leave off the cadmium reduction column and you can just measure nitrite directly. When you're done running the Feelizer, you need to make sure that you shut it off according to the instructions, that you put parafilm over your samples, and then store them in the fridge. And that's how the Feelizer works.